Hello, welcome to Lecture 9, Part 3 from Life in the Universe. In this video, we will conclude the material for this lecture, what we would have covered on our 9th class meeting. But this will not be the end of Unit C. We still have another lecture's worth of Unit C to do. In this video, we're going to look at some alternatives to some of the details of the biochemistry we've looked at in the previous two videos. So, any form of chemical life, it seems, is going to need something like DNA and also something like proteins. If it's not things made up of strands of amino acids, it's going to need some other way to put together complex molecules from simple components to do a lot of different things. And it's going to need something that's going to serve as a way to code for the sequence in which the components and its proteins or protein analogs will be put together. And even if we stick to just carbon chemistry, which we're going to do here, seems like there are clearly a number of alternatives. And one possibility is that there are other nucleic acids besides DNA and RNA, including some that use other sugars like threose, so-called TNA, and then do the chain by other than a phosphate group sugar backbone. So you could do that with peptides bonds, as we'll see here. So here are some alternatives. So the TNA just uses a different sugar as part of the base, but still connected by phosphate groups. That structure shown on the left. The B's are any of the bases that you might want to have there. And then here's the PNA, where you just have peptide bonds. So this little simple thing doing the backbone. And then here are a couple of other backbone possibilities, getting a little more far afield from something like DNA. But all of these potentially could work in the right circumstances, it would seem. Anyway, and also even with DNA, the way DNA is done by terrestrial organisms using just that selection of 4 bases on the DNA, that's not the only way you can do DNA. You can do DNA with other bases. There are pyrimidines and purines besides the ones we've considered. And it seems that besides the 4 bases that are used, it seems that there are at least 8 others along the way that purines and pyrimidines can bind to each other that would work for DNA. And so here's just some representation of that. So up here in the left corner, we see the 4 bases paired off with C and G and with A and T, the way DNA does it. But then here are some other pairs of bases that also would work. And some of these have names that only a biochemist could love, which we don't worry about what they're called. But anyway, as some example of some interesting experimental biochemistry, that bacteria have had their DNA modified to use one of these other, add an additional one of these base pairs in. The new base pairs don't code for anything typically, but the bacteria that have them can reproduce and survive just fine. If I'm not mistaken, if you're really interested, you look this up, see if I'm remembering this wrong. But I believe there actually has been a case of some bacteria with these non-standard bases where they have gotten the non-standard bases to code for polypeptides. One good thing about this kind of experiment is, are we concerned that we're now going to screw up life on Earth? These bacteria escape into the environment from the lab. And the thing is, the non-standard bases aren't found under natural conditions on Earth anymore. So at least not in current time. So they won't be able to get them to reproduce outside of the lab. But at least it shows that they work. Now, even with the genetic code we have, it turns out there are some slight variations in it. There are some cases where the RNA base code for different amino acids I told you about earlier in the previous lecture that code slightly differently than what the standard way. This is usually found in genes for mitochondria and chloroplast, which have been kind of separated from what is going on in nuclear DNA for a while. So it's maybe not surprising that that's where that variation would be. And also, even with the standard sequence there and the standard 20 amino acids, there are at least a couple of others that we know can occasionally be used. And for example, UGA, that RNA sequence is usually one of the stop codes. But if selenium is available, then it can actually be used to code for an amino acid called selenocysteine, which is normally not used in our amino acids, but cysteine is one of the ones that has a sulfur atom in it, and selenocysteine has a selenium atom where the sulfur was. And then there's another amino acid not part of the standard 20 that some archaea and bacteria will use UAG instead of a stop code to code for pyrrolysine, right? So this is an indication that the way most organisms do it on Earth isn't the only way it has to be done. And so presumably an extraterrestrial genetic code, even one using the same bases we use, could be quite different from the one that we see here on Earth. Okay, got a little bit more after this, but here is our attendant secret code. And that is the molecule phosphine. Chemical formula is PH3, a phosphorus atom and 3 hydrogen atoms. You might have noticed this has been in the news recently because phosphine has recently been detected in the atmosphere of Venus. And it forms not abiotically, which means not by biological processes, in the atmospheres of Jupiter and Saturn. But we haven't been able to figure out a way to form it abiotically under conditions on Venus. But there are bacteria on Earth that make it. So there are a lot of ifs here, but there's a region in the Venusian atmosphere where conditions would be, more or less okay for some pretty tough bacteria. So the question rises, could this be due to biological activity? At a minimum, this means there's going to be at least one more slide added from what I had planned when we get to the life in the solar system unit. But this is one of these things where a lot more work is needed. All right, I want to conclude this lecture, but not this unit. We've got more of Unit C to do in the next lecture, with one other aspect of biochemistry, and that is that asymmetric molecules like alanine, one of the amino acids shown below, have a handedness to it. Like our hands, one is a mirror image of the other. And for amino acids that have this handedness, basically amino acids can be abiotically produced either way, either in the so-called left-hand or right-hand versions. Organic chemists have a way of determining, of deciding which one they're going to call right-handed and left-handed. I'm not sure what it is. But the thing is, terrestrial organisms, when they go to assemble their proteins, use left-handed amino acids almost all the time. When they don't, when right-handed amino acids are slipped in, it's often in toxins. Another example of amino acids being used, say, or proteins, for example, being used in defense. And what makes them good if they have right-handed amino acids to act as toxins is because our biochemistry is generally oriented towards the left-handed ones. That means our bodies can have a lot of trouble dealing with the right-handed ones. And an example I know of an organism that uses right-handed ones in venom is the platypus. It might be something you didn't know about a platypus, which is one of the most absurd animals imaginable. I mean, it's a duck-billed, egg-laying mammal, and it turns out it has poison claws on its rear feet. Really, really odd creature. But anyway, this is important. So let's say that, tomorrow a flying saucer lands in front of you and some little green whatevers come out and say, hey, we're having lunch. Come on, join us. We got burgers. We got fries. Come on. It's a great lunch. Um, you Before you accept that invitation, find out if their biochemistry uses left or right-handed amino acids. Because if it's right-handed, if you eat a hamburger that was made from a right-hand amino acid using cow, you're not going to be able to digest it. You're not going to be able to break down those amino acids, and you are going to get very, very sick very quickly. So find that out. And by the way, if a flying saucer lands in front of you tomorrow and they're not friendly, not asking you to join them for lunch, but instead are shooting ray guns or whatever at you, you should know that it is a requirement of this course that if Earth is invaded by space aliens during the semester, it is a course requirement that you aid in the defense of Earth. So I probably should have put that in the syllabus, but anyway, we'll worry about that in when and if that happens. All right, so that's what I got you for this lecture. Speaking of food, my wife's been making dinner in a slow cooker all day. The house smells really good. So I'm looking forward to dinner, which should be coming along shortly. Anyway, I'll let you know when the Lecture 10 videos are